Bacterial infections are a huge problem. Bacteria become resistant to multiple antibiotics, and people have been imagining new ways to treat that problem. And phage therapy has emerged as a very compelling alternative to treat bacterial infections. Bacteriophages are viruses that find the bacteria, and then they enter the host and try and infect this bacteria. We thought, how does this interaction change under microgravity conditions? The physical forces impacting the bacteria and the bacteriophages are very different. How does the microbe respond to that? How do the phages respond to that relative to what we see under gravity conditions on Earth? People at the Department of Defense made us aware of an opportunity to run experiments in the International Space Station. And they said, well, here is your little box. You fit all your tubes into this little box, and that goes on a rocket. I was like, well, we can't do little boxes, we need more space. And they're like, no, this is what you get. You get a little box to do experiments. We were then put in touch with a company that facilitates this. And so my lab and this company, Rhodium Scientific, we said, all right, let's come up with an experimental plan. It was not something we had done before. Space is one constraint. Time of the astronauts is another constraint. The complexity of the experiments is another constraint. If we want to ask, how has microgravity impacted the biology of bacteriophage-host interactions, we have to have control conditions which exactly replicate the whole process, but on Earth, so that we can tell these two conditions apart. Once we sent the samples out, we were notified which rocket this was going to go up to the space station on. Phil Chudakarn and I, we flew over to Baltimore, and that we waited for the rocket to launch.
4, 3, 2, 1, and we have engine ignition.
And it was kind of surreal to imagine this tiny little box that was sitting on my lab's bench is now inside a rocket that's going to go to the International Space Station. The astronauts performed the experiments. We got the samples back and we ran analysis, and the results were really quite remarkable. Under microgravity stress conditions, both the bacteria and the bacteriophage pick up mutations that allow them to handle that condition better. And these mutations are really quite effective at killing pathogens on Earth. And so that relationship between what we might observe on the International Space Station versus what we see on Earth, they're not disconnected. There is a relationship there. And I don't think we definitively know what that relationship is, but that is really an invitation for us to explore that further.